Okay, thank you. So I will go through some of the machine learning methods that are used today in assessment of skin sensitization. So yeah, as was already mentioned, this is an important toxicity endpoint. And traditionally, in vivo test called LLNA is used to estimate the concentration of compound that makes 3 times proliferation of lymph node cells. And from this EC3 concentration, you can also estimate potency class of your compound. But now with the paradigm of with ban on animal testing, there is a need for new in vitro or in silico approaches, and these are usually combined together, and this is so-called a IATA, integrated approach to testing and assessment. And the selection of these new approach methods is usually based on the AOP, so adverse outcome pathway. And for skin sensitization, it's briefly, it's just there are 4 key events. So the first one is covalent binding to proteins, and the second is keratinocyte activation, and the third one is dendritic cell activation, which then leads to the 4th key event, which is T cell activation, and it results then in the adverse effect, so skin sensitization. And next slide, please. So we have today multiple so-called defined approaches. So these are defined testing strategies which have, it's important, fixed data interpretation procedure, and this is basically supposed to make them more regulatory acceptable because they try to avoid subjective expert judgment, and they provide clear rules how to assess your compound. And some of these methods, they also use machine learning, so they use data-driven methods to come to the final prediction that is based on multiple in vitro or in silico inputs. And we can go to the next slide. So briefly about machine learning. So basically, this is means that we build a mathematical model that takes inputs, so features, which in our case are results from in vitro tests or in silico predictions if we have them, and they try to predict target variable, which in our case is EC3 concentration or potency class derived from EC3. And this model is built on the training dataset. And here are a list of defined approaches that are used for skin sensitization. It is not a complete list, there are more, but I think these 6 are more, they are more reproducible if you look into the published papers, and they're relatively easier to reproduce compared to the others. And first 2 are really simple approaches, and they are not machine learning approaches, so they use simple rules. But the last 4 are actually machine learning, so they are based on the training data set and on the model that is built on this data set. And also these approaches can be distinguished whether they are used for hazard or risk assessment. So if they are used for hazard, it means they can only predict whether compound is sensitizer or non-sensitizer, but if they can predict risk, they can actually predict potency of compound. And I would just briefly go through the last 3 algorithms, so Bayesian network, neural network, and support vector machine. And Bayesian network is it's used to represent our probabilistic knowledge, and it means that if I know value of a certain variable, I can predict with a certain probability what is the likelihood for the value of another variable. And this network is built on our expert knowledge, and we use input features to predict the target variable. This is the potency class of your sensitizer. And one of the advantages of this approach is that it can also predict uncertainty. Next is neural network. So here they try to resemble the biological neural networks, and each node is supposed to be a neuron which can be active or inactive depending on the input it receives. And these activation functions for each neuron are determined on the training data set, and in the end you get a PEC3 value. So this method can be used for risk assessment. And the last one is a support vector machine. So here this is a classification algorithm for classification task, and it can distinguish sensitizers and non-sensitizer based on the input features that are provided. And it finds the optimal hyperplane. So this is just a sketch in 2D, but in reality we have more features and it's difficult to visualize. And next I have a brief comparison of these different approaches. So first, if we compare how they perform for the LLNA hazard, so we compare these different defined approaches, and we can see that they have comparable performance if we look at the accuracy. But if we look also at the sensitivity and specificity, we see that Bayesian network and support vector machines are maybe more balanced than the neural networks, which have low specificity, meaning they will get more false positives. And next, we can compare also these results for together with LLNA to predict the human hazard, and we can see that these defined approaches that use machine learning, they have comparable or better performance than LLNA. And also, if we continue, I have also 2 slides for comparing the risk assessment. So here we compare Bayesian and neural networks, how well they predict potency, and they have comparable accuracy. Of course, the amount of over or under predicted compounds is then be different, but the accuracy is comparable. And if we add, go to the next slide, here we compare together the machine learning approaches with LLNA, how well they perform to predict human potency, and we see that actually their performance, their accuracy is comparable to the LLNA. So to the next slide, maybe. So here it's just summary of this comparison of different defined approaches. So advantage of Bayesian and neural networks is that they can be used for risk and hazard assessment, while support vector machines is only used for hazard assessment. And the advantage of Bayesian networks is that they can also predict uncertainty of the result, while neural networks and support vector machines cannot. But all 3 approaches maybe lack some interpretability. It's difficult to interpret these models, and they're also difficult to implement on your own. But next slide. So here are just conclusions, and we saw that all these defined approaches that use machine learning are comparable or superior to the LLNA, and they require specific knowledge if you want to implement them. But luckily, for example, our Safer Skin application makes it easy for user to apply Bayesian network. So this is why I think one good example how you can still apply this relatively difficult to implement defined approach. And maybe this lack of transparency of these models can be a barrier for regulatory acceptance, and it's up to discussion, but maybe some like hybrid approach is more appropriate than just pure data-driven algorithm. So with that, I would like to thank you for your attention, and I would be happy to answer your questions at the end.